Welcome to today's video. Today we're going to be talking about cancer as a metabolic disease. This is opposed to the more traditional historical view of cancer as a genetic disease, where the historical idea has been that outside factors from the environment, such as radiation or chemical mutagens or some things like asbestos, will cause mutation to the genomes of your cells, and eventually the genomic damage, the damage to the cell, will accumulate so much so that cancer cells develop from this. Now, this is the idea or the concept that modern medicine, modern medical treatments derive their treatments against cancer from. And it's also one of the reasons why none of the treatments against cancer try to attack the underlying cause of cancer according to the genetic disease model of cancer. Because if the genetic diseases or if the genetic mutations were truly the underlying cause of cancer, it would, given current technological knowledge, current biological tools, be practically impossible to completely revert all the mutations that occur in the various cancer cells in a cancer patient. So it simply currently isn't an option to completely eliminate all the mutations. And because of this, the modern treatments, which of course derive their approach against cancer from the genetic model of cancer, aim not at fixing or curing the underlying cause of cancer, but rather they attack the symptoms of cancer, which of course are the abnormal growth, the metastases, and the displacement of other tissues. And this is where chemotherapy, radiation therapy, and immunotherapy all come in, all of them through various means try to curb the growth of cancer cells. But of course, in doing so, because they are all not specific, so none of them target cancer cells specifically, they all also attack the cells of your body, the healthy cells, some of which are fighting cancer cells actively, like the immune cells. And of course, this can cause major side effects. One of the most well-known is probably the loss of hair in chemotherapy patients, but also things like complete obliteration of the immune cells are quite common. Now, beside these negative effects of the treatments, there's also a few fundamental issues with the genetic model of cancer, a few studies or a few phenomenon that the genetic model, called the somatic mutation theory, cannot really explain. So this is kind of where the metabolic theory comes in. It adds on to the somatic mutation theory, and it takes a different approach to the cause of cancer, not actually being the genetic mutations, but rather the primal cause being an abnormal metabolism and damage to the mitochondria. So this theory is called the mitochondrial metabolic theory, and we'll of course go into greater detail on that later. And it tries to explain phenomena that the somatic mutation theory, the genetic model, can't explain. And of course, this is one of the main things we'll be talking about today, is how do these models differ? How do they explain, or how do they fail to explain, some of the things we see with cancer cells? And of course, what are the implications of the different models for our options for treatment? Because, as a little spoiler, the metabolic mitochondrial theory, the metabolic model for cancer, would allow more root cause treatment of cancer. It would allow us to metabolically and dietarily and through lifestyle choices, to attack the underlying cause of cancer, which of course hasn't been a possibility when working under the genetic model. So that's basically the plan for today's video, for this, I guess, little lecture. Before we dive into that, though, a bit about myself. So I'm Cedric, and I'm one of the co-founders of Marchwood. We help cancer patients, such as perhaps yourself, with outliving cancer by kind of parsing and digging through literature based on our previous experience from the Bachelor of Science in Biotechnology, which both my brother and I have. And we kind of look through the literature at the newest findings on cancer and potential cancer therapies, and we do our best, of course, to explain this to people like you, so that you can understand your disease and so that you can outlive your disease more successfully. Now, we've also got a community project that we are currently building. It's currently still in its early phases, But if you are interested and if you are yourself a cancer patient, you want to connect and socialize with other cancer patients who are going through the same struggles as you, and if you want to learn more about your disease so that you can outlive it, you can head over to our community project at marchwood.com/outlive, where you can then join us. And of course, we'll also be in that community, and we'll be helping out and be open to any questions that you might have. Now, with that out of the way, we can dive in, and we'll begin with the somatic mutation theory of cancer. Now, this is the traditional theory of cancer, and it basically posits that if you have a normal cell, just a completely normal cell in your body, that it will transform into a cancer cell, which, for the sake of this video, just so you have a visual differentiator, I will make all the healthy cells round, and I will make all the cancer cells, rectangular. And will transform, a healthy cell will transform into a cancerous cell by mutations. So basically, you have a completely normal healthy cell, mutations occur, and will become a cancer cell. Now, the place that these mutations occur in is the genome. So if you look at the graphic of a cell that I drew, you have the cell membrane, which kind of delineates the cell from everything outside of the cell. So cell membrane is kind of like the walls of your house. And inside of the cell, you have something called the cytosol, which is kind of just let's call it a cellular soup. Even though that's, of course, not the technical term, I think it gets the point across. And this is where a lot of the cellular components kind of float around in, and they move about. This is, for example, where proteins that are currently being synthesized could be. This is where mitochondria, which we'll get into later, kind of soup around. And it kind of allows transfer of different metabolites and different things within the cells. Of course, it's basically just a water soup with a bunch of biologically active components. Now, another major component that we have in the cell is the nucleus, and this is where This is where the genome resides. So anytime there is something about the specifically somatic genome, so the genome of the cell, it will be inside of the nucleus, which will become quite important later on. And the argument of the somatic mutation theory, the genetic model of cancer, is basically that the mutations impact and destroy this genome so much that it kind of becomes either completely mutated or that the chromosome count might change, that basically the genome that in your normal cells regulates the normal cell behavior. So the genome has, of course, many downstream effects, proteins which are synthesized, how active those proteins are, and how exactly your cell behaves. A lot of that is based on the genome and the genetic regulation. And the argument is the mutations destroy the genome, causing completely different regulation and thus also different growth regulation, which leads to cancer cells proliferating. Now, the problem, one of the main problems and one of the experiments that shows this is that the genome itself doesn't actually seem to be causative of cancer. So a very good experiment that showed that is the so-called nuclear transfer experiment. This experiment basically said, okay, we now have healthy cells and we have cancer cells. And we believe that, or the somatic mutation theory posits that this genome and the nucleus thus is the cause of cancer. So they basically said, okay, this is the cause of cancer according to the somatic mutation theory. So what would happen if we took a completely healthy cell and we, instead of leaving a normal genome inside, took a cancer genome from a cancerous cell and transplanted it into the healthy cell? So now we have a healthy cytosol, the healthy cellular soup, and a completely normal cell, except for a cancerous genome. According to the somatic mutation theory, this should result in a cancer cell. Because, of course, the somatic mutation theory says that the mutations are the primary cause of cancer, and that thus, if we transfer the nucleus and thus also the genetic mutations into a healthy cell, this should upregulate cell growth and cause this cell to become a completely normal cancer cell. But interestingly enough, I'm gonna mark this in red, this is actually not what the experiment showed. What it showed instead was that such a cell actually maintains completely normal cell behavior. So the genome which has been mutated, of course, it doesn't revert. The cell does not have the ability to completely go back to its unmutated genome. So the genome maintains its mutated state. However, the cell itself will behave like a completely normal cell. So this is basically goes completely against the expectations of the somatic mutation theory. And that means that the genome alone and the genetic mutations cannot be the sole cause of cancer. There has to be something in the cancerous cytosol, in the cancerous cell, outside of the genome and the mutations that cause this behavior, because apparently only transferring the genome and thus the mutations isn't enough to induce cancerous behavior. Now, if we just go one further, there is another very interesting thing that this same experiment, I guess, found out. So, there we go. So, what this research also looked at is it took a cancer cell. So we have a cancerous cell, and they said, you know what? Before we had a healthy cell with a cancerous genome, let's try the opposite. Let's actually take a cancerous cell and let's put a completely normal genome and nucleus inside it. Somatic mutation theory, again, in this case, says, okay, cell behavior and thus cancer behavior is dependent on the genome. This should, according to the somatic mutation theory, according to the genetic model, yield a, you guessed it, it should yield a healthy cell. This should yield a normal cell according to that model. However, you might already be getting or be understanding what I'm getting at. This is once again not the case. Once again, what we actually see is the exact opposite of what we expect. A cancerous cell with a completely healthy and non-cancerous genome will actually lead to a cancerous cell with cancerous behavior. And interestingly enough, it will also lead to a cancerous nucleus. So now all of a sudden, it seems like the opposite is actually the case. It would seem, based on these results from the nuclear transfer experiment, that it is not the genome that dictates whether a cell becomes cancerous, but rather the cytosol surrounding it. Now, the interesting part of this is, of course, also that in a cancerous cytosol, so if you take a cancerous cell and place a healthy genome inside of it, you still get these mutations. So it does seem that these are in some way centrally important to a cancer being a cancer cell. And it's also considered one of the hallmarks of cancer, because simply having a somewhat dysfunctional cytosol isn't necessarily enough for something to be considered cancer. So it is not so much that the somatic mutation theory is completely wrong. It is not that the mutations that we see in cancer are completely irrelevant, but it is simply that it seems that it is not the mutations that cause cancer. So it doesn't seem to be doing this does not seem to be the case. It does not seem to be that mutations lead directly to cancer, but rather it seems that the write this up. leads that the cytosol leads to cancer on its own, but also that it leads to mutations, which are a downstream effect. Sorry for my poor handwriting. I'm somewhat not used to writing at this angle, but that the cancerous cytosol leads to mutations, and it is kind of a cofactor in cancer. So the fundamental underlying cause based on this isn't the genome, but rather it is the cytosol, and this is where the new theory, the so-called mitochondrial metabolic theory, comes in, which takes these findings and a few others and basically says, look, we're going to posit that the fundamental cause isn't the genome, but that the fundamental cause is abnormalities to the mitochondria, and by extension also to the metabolism. So I'm going to go to the next page again, and then we're going to look at what exactly that means. Alrighty, so in order to understand the mitochondrial metabolic theory, we have to look at what exactly mitochondria are. Now, if we once again draw up an image of a cell, you know, of course, now that it has a wonderfully drawn genome and a nucleus which contains this genome. And of course, you know about the cytosol. Now, within this cytosol, there are further small compartments that are called mitochondria. These are there we go. And these mitochondria, they are small subcompartments, so they are spatially distinct from the rest of the cytosol, and they also have a membrane that delineates them from anything else in the cells. And this is what we call small organelles. So basically a small subcompartment, like a room inside of your house. And these mitochondria, they're important for a plethora of things, but one of the major, I guess one of their major, what's it called? I know the German word, but for some reason the English word responsibilities. There we go. One of the major responsibilities of this is oxidative metabolism. Now, this is metabolism that involves oxygen, thus the name. So basically any metabolism that involves oxygen for the production of cellular energy is called oxidative metabolism. And all of this oxidative metabolism occurs inside of the mitochondria. Now, beside this, which is what most people know about, some of you watching this video might even have heard of the mitochondria as the powerhouse of the cell. What few people know is that the mitochondria actually also have a different function, which is, I will call it quality control. So they basically have the ability to keep tabs on the genome and on the rest of the healthy cell, and they can see how healthy is this cell. Is it healthy, or is it maybe dangerous to the organism? If it is dangerous, so if they see that there is something that is horribly wrong inside of the cell, or maybe inside of the genome, for example, maybe they see cancerous genetic mutations, then the mitochondria have the ability to induce something called apoptosis. So apoptosis is basically the fancy term for cellular suicide, and the mitochondria have the ability to look at the cell, realize that it is defect, and then induce cellular suicide to ensure that the cell doesn't become dangerous and threatening to your body. But of course, cancer cells, which are dangerous, damaged, and threatening to you as an organism, they don't do this. There is something that happens in cancer cells that inhibits this induction of cellular suicide by the mitochondria. And this is because cancer cells have abnormal mitochondria. Their mitochondria are damaged, and they're no longer able to properly perform the actions that you would see in healthy cells. So Whereas with normal cells, you might see completely normal mitochondria. In a cancer cell, we go to the next slide. So in cancer cells, you would instead not see completely normal mitochondria, but you might see some deformed, abnormal, and dysfunctional mitochondria. Now, this damage can stem from a lot of places, but one of the main ones are dietary and lifestyle factors. So the metabolism of your organism, what you feed it, significantly impacts mitochondrial health. What you eat also is known to significantly impact mitochondrial health. And in cancer, these abnormal mitochondria lead to an uncoupling and damaging of the mitochondria. And this means that the mitochondria don't fulfill their roles. They don't quality check the cells and the health of the cell, and thus the cells end up being able to become cancerous in the first place, and the mutations can accumulate without the cancer cell being stopped. Now, there are a few ways by which this damage to the mitochondria can occur, or there are actually quite a few, but of course going into all of them in exceptional detail would be impossible for a reasonably length YouTube video, right? We don't want this to end up being a 20-hour video where we go into all the details. But one of the main ways that this can happen is glycolytic metabolism. Now, this is metabolism that occurs whenever your body has sugar available. So whenever there is a lot of sugar inside of your body, your cells will metabolize it, and this is termed glycolytic metabolism. Now, it is somewhat complex because the glycolytic metabolism, the glycolytic part of this pathway, actually occurs within the cytosol. This is a metabolism that does not need oxygen. So the glycolysis itself does not need oxygen, and it occurs within the cytosol. However, because of course it occurs within the cytosol, this means that the mitochondria themselves are not needed for a purely glycolytic metabolism. Of course, you can also have the downstream, you can have some downstream molecules from this glycolytic metabolism that can enter the mitochondria and be metabolized further utilizing oxygen. But when you feed your cells a lot of sugar, a lot of glucose, they will simply rely on the glycolytic metabolism more heavily instead of also utilizing the oxidative capacity of the mitochondria. Furthermore, the glucose itself, so the glucose that is needed for this glycolytic metabolism, can also directly cause damage and stress to the mitochondria. Furthermore, the reliance on glycolytic metabolism can indirectly make your cell and also your mitochondria much more susceptible to toxic and oxidative damage and glycative damage from the outside by inhibiting something called the glutathione system. Now, this is a system that exists within your cells, and its main purpose is to protect your cells, your DNAs, and your structures from toxic damage, from glycative damage, that is, damage that happens when sugar molecules randomly attach themselves to proteins or to DNA or to some fatty acids. So basically, random sugar addition is glycative damage, but also from oxidative damage, which happens when reactive oxygen species, this is a type of oxygen that is highly reactive, reacts with random structures in your cells. So maybe the oxygen just randomly oxidizes proteins inside of your cells. And of course, it is a very fine balance. So if you start randomly destroying and randomly changing the structure of the proteins and of the molecules and of the DNA in your cells, there's going to be a lot of side effects and a lot of things will start functioning significantly less. And to protect your body from these random side effects, the glutathione system basically acts as a buffer. So what happens is that you have something called reduced glutathione. So this is kind of the unactivated, unutilized state of glutathione, and it can transfer into oxidized glutathione. So these are two types of you can see the GS, that kind of stands for glutathione. And then here you have two glutathione coupled together, GSSG. And in this kind of transformation, to put it very mildly, it can absorb damage. So if you have glycative stress, so a lot of sugar floating around in your body that wants to randomly add itself or attach itself to proteins, to other molecules, or if you have oxygen, reactive oxygen species that are highly reactive and might react with random components inside of your cells, the glutathione system, this reduced glutathione, has the ability to instead act as a absorbent, kind of like a shield. And of course, as you can see now, at some point this glutathione, if you place a lot of stress on the cell, will run out. Now, the good thing for us, and one of the reasons why we can live for as long as we can and we don't die once the initial glutathione runs out, is because our bodies can restore reduced glutathione. And this mainly happens in the mitochondria. So our bodies kind of use this glutathione to absorb stress and damage, which would otherwise ruin the cell itself. And then they restore the glutathione to its reduced format, where it is then once again available to be used as a shield. The problem with this glycolytic metabolism that occurs, for example, with high carbohydrate diets, is that this restoration of the reduced glutathione, the glutathione that is needed to absorb damage, let's actually use the color red, that this is inhibited, which means that at some point the glutathione that your body has built up runs out, and then all of a sudden your body has much less of a shield against random stressors, which of course can lead to these stressors attacking the genome. So once the glutathione runs out, you will be much more likely to incur genetic mutations, which can of course explain why earlier we saw that a cancerous cytosol with cancerous mitochondria and thus also dysfunctional glutathione system led to significant mutations in the genome, because now the glutathione system can no longer protect the genome from random stressors, from glycative damage, from oxidative damage, and thus the mutations occur. Now, furthermore, the mitochondria, as this is biology, are very complexly regulated themselves. So there are a lot of regulatory pathways, I will call them. This is kind of downstream effects where proteins expressed by the genome affect other proteins, and then other proteins is kind of like a chain of command, or a chain of command is kind of a good way to put it, where the genome will affect the mitochondria through the expression of various genes. However, this isn't a one-way street, and the mitochondria are also very much involved in regulating the genome itself. And because of this, the suppression of the mitochondria by over-reliance on a glycolytic metabolism can lead to genome instability. It can lead to differential gene expression, where genes that usually make cells susceptible to induced suicide, to this apoptosis we had earlier, where they become downregulated, and where genes that protect them from this induced suicide are upregulated. And of course, this means that when your body, even when immune cells recognize that, hold up, this cancer cell is dangerous and we need to do something against it, that this cell has now become much, much more resistant to your body basically telling it, hey, you have to kill yourself because you are a danger to us as an organism, and if you continue to grow, we will die, right? This is normally what happens. And all of these are downstream effects of abnormal mitochondria. Now, the good thing for us is that, as you can already see here with the glycolytic and oxidative metabolism affecting mitochondria, it allows for metabolic intervention. It allows for dietary intervention because the mitochondria are centrally important to metabolism. If we can somehow take a cancer cell with its cancerous genome and its cancerous mitochondria, if we are somehow able to reform these mitochondria into completely functional and completely normal mitochondria, we can actually restore either the apoptotic signaling, where the mitochondria will enforce cell suicide upon the cell, or we can restore normal cell behavior. So basically, the genome, as you can see, the genome, and as you can also imagine, the genome, like I said, it is not going to be healed. Just like we saw in the nuclear transfer experiment, where you transferred the nuclear genome into a normal cell, the genomic mutations simply remain. There is no real way to heal them all, except for specific interventions in the laboratory. But even so, we have at the moment no ability to do such a thing. However, despite this, the mitochondria, when they are rehabilitated, when they are reactivated, and when they are healed, they have the ability to enforce normal cell behavior onto a genome, even though it is cancerous. And this, of course, opens up new options of treatment, whereas previously we thought we only had the genome as an angle of attack, which left us with only symptomatic treatments against cancer. We now all of a sudden have a completely new avenue and new ways of dealing with cancer, which means that we can now look at dietary interventions. Now, as we said before, one of the major drivers of mitochondrial impairment, of this damage and these abnormal mitochondria, is the glucose metabolism. So you might now be thinking, okay, if that's the case, maybe cutting out sugar from your diet, right, should reduce how much sugar gets to the mitochondria. It should reduce the inactivation and the damaging of the mitochondria. And it should thus also lower the cancer behavior of cells. Now, of course, this isn't going to be perfect. This is biology we're talking about. This is never a 100% thing in biology. It's always all about balances, and it's always about equilibriums. However, research has shown, there's a lot of promising research that has looked into simply feeding cancer patients ketogenic diets. So, for example, these are diets that simply ensure that your body switches from utilizing sugar commonly to utilizing something called ketones and fatty acids. Now, in doing this, in entering a ketogenic diet, the average blood sugar over extended periods of time in your body will drop, because of course you're no longer eating carbohydrates which spike your blood sugar, which then again means that the blood sugar and thus the sugar available to the mitochondria and the cells is significantly lower. This means that the cells themselves will switch from the glycolytic non-oxidative metabolism to a metabolism that relies on fats and ketones. These are, of course, you know what fats are. Ketones are fat-derived particles that are metabolized in much the same way. So they are simply, ways that your body can transport energy or energy fuel throughout the body and disperse it to the various tissues of your body, besides the fatty acids, which it of course has, and the blood sugar. And of course, when it switches to that, this oxidative metabolism happens within the mitochondria. So a ketogenic diet basically forces cells to rely heavily on the mitochondria, which means that they will become reactivated. And it also means that there will be less damage to the mitochondria. And in the end, research, like I said, has shown that this leads to significantly improved outcomes for cancer patients, just like the mitochondrial metabolic theory would suspect. Furthermore, there are other things, for example, acidification of the tumor environment. There are many, many things that impact how exactly the mitochondria, how they function. And by attacking the various causes of mitochondrial dysfunction, if they are rehabilitated, this leads to completely this will lead to normal cell behavior, and it will allow us to kind of utilize the mitochondria to fight cancer and to attack cancer at its root cause, which is something that wasn't possible if you simply follow the somatic mutation theory. And this is basically also what our method is based upon, the Mosaic method, which my brother and I developed. It is based on this theory, and then it includes, of course, various, scientific literature from various different subfields of molecular biology, and it pulls from various different places to look at what options, beside, for example, the ketogenic diet, which I mentioned, what other options do we have to help our mitochondria inside of the cancer cells normalize, so that we now have the ability to attack cancer cells at their root cause, which means that given this, we could actually cure cancer. Now, another great thing of improving mitochondrial health is that it doesn't negatively affect the other healthy cells in your body, which is, of course, something that I spoke of before with regard to chemotherapy and immunotherapy, radiation therapy. All of those nuke the body and attack all kinds of healthy cells. And the benefit about mitochondrial rehabilitation and aiming to restore the mitochondria in cancer cells is that in the normal cells, or in normal cells with a normal genome as well, for example, in your brain cells, in your immune cells, in your hair follicles, in your mucosa, all of these cells which are normally heavily attacked by chemotherapy, by traditional treatments, they all have normal mitochondria if they're not cancerous. So if we treat cancer by normalizing the mitochondria, all of these cells will be unaffected by the treatment, which means that the side effects are significantly lower, up until non-existent at all. For example, a ketogenic diet. There are many people who go onto ketogenic diets for years on end without eating any carbohydrates or any significant amount of carbohydrates, and they are doing completely fine. Now, if you had someone go onto intensive chemotherapy for years on end, I think we could both agree that they would not be fine. Now, of course, I think it would blow the frame of this video a bit to try and dive into the details behind all of the various approaches, because all of them affect the mitochondria and the cells slightly differently, and it would take much, much too long to attempt to explain the various details of what options we have for normalizing the mitochondria. But if you're interested in learning more, we've actually written up a document called the Mosaic Method Guide, which goes through the scientific and molecular biological basis behind what steps we recommend to cancer patients, because there are some steps that we recommend to any cancer patient which are simply recommendable because there are basically no side effects to those steps. This is, for example, a ketogenic diet. It would include something called alkalization, astaxanthin, and beside all of these various interventions, how exactly you could implement it into your life, this Mosaic Method Guide also explains why we choose them. Why do we recommend a ketogenic diet? And, which might also be interesting for you, is that it includes all of the scientific literature that stands behind our reasoning. It includes all of the citations that we base our recommendations upon, which of course allows you to go in there yourself and allows you to check that we are not just spewing, complete nonsense. Because of course, we are people on the internet. You never know with random people on the internet, but both my brother and I, we believe in transparency, we believe in honesty, and we believe in educating people. So we basically want to say, look, here are all of the studies, all of the scientific literature that we base our suggestions on, and then we want you to come to your own conclusions, whether that is sensible or not. We don't want to fool you into something, and we basically want you to be able to make your own informed decisions, even if that decision may end up being that we are complete and utter clowns. We want to enable you, and we don't want to just fool you into something, which is why we've put together the Mosaic Method Guide. Like I said, you can find that under marchwood.com/mosaic. If you head there, you can download it, and then you can check out all the scientific literature that is behind this, all of the other videos we've done on the channel, and of course also behind the method that we suggest that people do, behind the interventions. And yeah, that's basically all I had to say in this video. I hope it was very helpful. I hope you kind of got an insight into the mechanism behind cancer, how the mutations that occur in cancer and how the metabolism of cancer cells are actually intrinsically tightly connected, and that you now might have a greater understanding of what you can do against your cancer. So yeah, with all of that said, I hope you have a wonderful day. I hope you swift healing and lasting health. Goodbye.